Have you ever stopped to think about how your brain just knows the difference between, say, a tiny bird's chirp and a huge lion's roar? I mean, it seems automatic, right? But it's actually one of the most incredible things our brain has to learn. Today, we're going to dig into the science of how our brain literally builds its own internal map of sound and how experience tunes that map to perfection.
Seriously, just think about it for a second. The piercing scream of an alarm clock, the deep rumble of thunder. Your brain processes this insane range of sounds instantly. But how does it do it? Well, the secret isn't just in your ears. It's actually in this beautifully organized, almost elegant map that's built deep inside your brain.
And this organized map, well, that's our first big clue in this whole investigation. See, it's not a map of places, like a city map. It's a map of pitches, a sound map. And figuring out how it gets built is the key to solving the whole mystery of how we learn to hear in the first place. Now, the official scientific name for this is tonotopy. I know it sounds super complicated, but the idea behind it is actually really cool and simple. All it means is that the neurons in your brain that get excited by similar sound frequencies are physically located right next to each other. So you've got your high-frequency neurons in one neighborhood, your low-frequency neurons in another, all laid out in perfect logical order. Okay, the absolute best way to picture this, just imagine a piano keyboard laid out right across the surface of your brain. You've got the low notes on one end, the high notes on the other, and everything in between arranged in a perfect gradient. That's pretty much what your brain does. One end of this neural map handles the deep rumbles, and the other end takes care of the high-pitched squeaks. Right, so this idea of a perfectly tuned piano in our heads brings up the huge question that scientists have been wrestling with for years. Are we just born with it? Is it hardwired from day one? Or does our brain have to learn how to tune this piano after we're born, based on all the sounds we hear? That is the mystery we're going to solve. And you know, in any good mystery, you've got to have a prime suspect. To figure out if this sound map is learned or just built in, scientists zeroed in on one very specific molecule, a tiny protein that was already a known superstar in brain development. And that suspect is brain-derived neurotrophic factor. Let's just call it BDNF, because that's a mouthful. You can basically think of BDNF as fertilizer for your brain. It helps your neurons grow, survive, and build strong connections. It is absolutely essential for what we call brain plasticity, which is just the brain's amazing ability to change and adapt based on experience. So the hypothesis, the big idea, was pretty straightforward. What if BDNF is the molecular construction foreman on the job site? What if it's the one that's walking around, giving out instructions, telling each neuron which key on that piano it's supposed to become? Maybe it's the key to creating that perfect sound map.
Now, a good idea is one thing, but in science, you've got to test it. So to see if BDNF really was the foreman, researchers designed this really elegant experiment. And it's a classic setup, a tale of two very different groups of mice.
So here's the breakdown. On one side, you've got your wild-type mice. These are the control group. They're just normal mice with normal levels of BDNF. But on the other side, you have these mice that were genetically engineered to produce about 22% less BDNF. They're our test group. The idea is, if BDNF is really that important, we should see some major differences in their brain sound map. And wow, the results were incredible. First, the researchers looked at the physical structure of the neurons themselves. And in the normal mice, they saw that neurons that process high frequencies were built differently than the neurons that process low frequencies. But in the mice with less BDNF, that critical difference was just gone. The biological blueprint was totally scrambled. And that structural problem created a huge functional problem. Because in the normal mice, the neurons were tuned differently, just like the keys on a piano. But in the mice with less BDNF, all the neurons were tuned the same way. It's like every single key on the piano played the exact same note. The whole map was broken. But wait, this brings up another giant question. So the map is broken, but maybe, just maybe, the brain can fix it with practice, right? What if you just exposed the brain to more sound? Could it learn to overcome this biological problem on its own? This question led to the most important part of the whole experiment. The logic here was simple but so smart. They took the mice with less BDNF, and during this really important window in their development, they put them in an environment that was just filled with all kinds of different sounds. The big question was, could all this extra sound experience, all this practice, force the brain to tune its piano correctly, even without enough of that BDNF foreman molecule? And the answer was a clear, definite no. As the study put it, The mice with BDNF reduction were unable to properly respond to changes in sound-evoked activity. What that means in plain English is that even with all that extra sound stimulation, their brains just couldn't use the information. They couldn't adapt, and they couldn't build a proper map. This was the smoking gun. It proved that experience by itself is not enough. You need both ingredients. You need the sound coming in from the outside world, sure, but you also need the right molecular tools, like BDNF, on the inside. Without that BDNF foreman on the job site, the brain's construction crew just can't build the map, no matter how many blueprints you throw at it.
Okay, so we know the map gets totally scrambled without enough BDNF. But let's get real for a second. What does that actually mean for the mouse? What's it like to hear the world through a brain that's so out of tune? Well, that's exactly what the researchers wanted to find out next.
So to figure this out, they played a very specific high-frequency tone to both groups of mice and watched what happened in their brains. And the difference was just stunning. In the normal mice, only the correct high-frequency neurons lit up. It was a clean, targeted, precise response. But in the mice with less BDNF, neurons started firing all over the map. It was a chaotic, disorganized mess of noise. And this right here is the real-world consequence. Without that organized map, the brain literally can't tell one sound frequency from another. A single, clear, high-pitched note just becomes this chaotic explosion of neural activity. It's not just that the piano is out of tune. It's like someone ripped all the keys off and just glued them back on in a completely random order. There's no way to make sense of the music.
So, what's the final verdict here? What are the big takeaways? Well, number one, our ability to hear isn't something we're just born with. It is actively built after we're born. Two, this whole building process needs a partnership between the sounds we hear and this amazing molecule, BDNF. Three, if you don't have enough BDNF, that sound map gets scrambled. And maybe most importantly, experience alone can't fix a brain that's missing its master builder.
And all of this leaves us with one last pretty powerful thought. This whole study is just a fantastic example of neuroplasticity in action. It's stone-cold proof that our experiences physically shape our brains. So if the sounds you hear every day are constantly sculpting the parts of your brain that hear, it kind of makes you wonder, right? What other daily experiences, the things you see, the things you think, the things you do, are sculpting the rest of your mind right at this very moment? Definitely something to chew on. Thanks for joining me for this deep dive.